Get comfortable and let your breathing slow. There's nothing left to do tonight but rest. Let the day fade as we settle into the quiet drift of tiny particles falling through the deep ocean. If this brings you rest, subscribe, and there'll be a new one waiting tomorrow night. Below the surface of the ocean, where the last of the daylight has not yet given up entirely, the water is full of falling, not the kind of falling that rushes or that carries any sense of urgency. This is something else, something patient. Individual particles drift downward through the blue at the pace of dust settling in a still room, slowly enough that a single particle might cover no more than 5 meters over the course of an entire day, slowly enough that it seems, for long moments, to be going nowhere at all. The surrounding water is very cold. The light that filters down from the surface arrives in diffuse, shifting columns, pale gold, giving way to pale green, giving way to a blue, so deep it has almost become another word for dark. And through that blue, through those slow columns of dissolving light, the particles fall. They are small. Most are far smaller than anything the eye could name, and the few large enough to see look pale and loosely structured, almost transparent, faintly luminous, where a last trace of surface light catches their edges. They drift, they rotate very slowly, nudged by currents too gentle to feel. They appear at first to be nothing. They are not nothing. Each one is a ruin and a record and a small engine of transformation all at once. The ocean's sunlit surface is a place of furious, invisible production. Phytoplankton, cells so small that a drop of seawater may hold 1,000,000 of them, spend their brief lives drawing dissolved carbon from the water and building it, with the energy of sunlight, into living tissue. They divide, they bloom, in the right condition, in spring and summer across temperate and polar seas. They build themselves up in such numbers that the surface water turns green, or red gold, or pale turquoise depending on the species, and the bloom can be seen from space as a slow stain spreading across the surface of the ocean. And then they die, or are eaten, and the eating produces more fragments, and the fragments aggregate, and what comes next is the subject of everything that follows below. A phytoplankton cell, at the very end of its life, begins to lose integrity. The membrane that has held it together through division after division softens, and the contents start to leak, and the cell collapses slowly inward on itself, releasing fragments of its structure into the surrounding water. Nearby, other cells are doing the same. Between them, clinging to their surfaces, and free-floating in the spaces between, is something that cannot be seen, even with the naked eye, a substance without color, without form, almost without substance in the conventional sense, and yet perhaps the most important material in the entire process of what is about to begin. Phytoplankton and bacteria, as they live and as they die, release into the water around them a class of gel-like, sticky material. These gels form threads and films and loose webs invisible to any ordinary observation, spreading through the water between particles that would otherwise simply drift past each other without touching. When a fragment of dead phytoplankton encounters one of these threads, the thread holds it. When another fragment drifts into contact with the first, the same gel holds it too. Slowly, without any directing force, without any organism planning or coordinating the process, loose particles begin to gather into something larger. They accumulate the way sediment accumulates at the bottom of a glass of cloudy water, not by choice, not by design, but because the physical and chemical conditions make accumulation the natural outcome of contact. One fragment becomes two bound together, two become five, five become a cluster visible, at its largest, to a careful human eye. An oceanographer working in the early 1990s first characterized this gel-like material as its own distinct class of particle, transparent exopolymer particles, though the name matters less than what they do. They are the biological glue that makes marine snow possible. Without them, the fragments of dead phytoplankton would remain fragments, suspended and small and slow to sink. With them, the fragments become aggregates, and the aggregates are what fall. The name came from the appearance. A researcher with a submersible's lights turned into a water column full of these drifting aggregates would see in the beam what looked like snow, pale, unhurried, moving in the same loose, wandering way that real snow moves through still, winter air. Marine snow. The name is accurate in its impression and a little misleading in its implication of cold and winter, because marine snow falls in every ocean, in every season, without pause, in waters tropical and polar alike, at every hour of every day. It is not an event, it is a condition. The ocean is always snowing downward into itself. What goes into the snow is not simple. A single marine snow aggregate, examined closely, contains the remnants of many lives and many processes, all bound together by gel and chance contact and the slow logic of aggregation. There are fragments of phytoplankton, the silica shells of diatoms, still holding, in many cases, the ghost of their intricate geometric structure, their patterns of pores and spines, intact even weeks after the cell that built them has been consumed. There are fragments of the soft, unstructured cellular material of other phytoplankton types, formless now, recognizable only by their chemistry. There are bacteria, living ones, colonizing the outer surface of the aggregate and working at the organic material within it. There are mineral fragments, tiny grains of clay or silt, carried into the ocean by rivers and wind, that have settled down through the surface layer and been incorporated into an aggregate simply by contact, by adhesion, by the same gel threads that hold everything else together. And there is something else, something that, in terms of its influence on how fast the aggregate sinks, matters more than most of the rest. Among the animals of the ocean's sunlit surface, the copepods, the krill, the salps, the small translucent creatures that spend their lives grazing on phytoplankton, eating produces waste. This is not incidental. The fecal pellets produced by these small animals are remarkable objects in the context of what falls through the ocean. A copepod, which is an animal a few millimeters long, with a paired set of antennae and a jointed body that rows itself through the water with steady mechanical precision, produces pellets that are cylindrical, membrane-bound, and dense in a way that fluffy phytoplankton aggregates are not. Where a loose aggregate of dead cells and gel might take weeks to sink a few hundred meters, a fecal pellet from a copepod might sink the same distance in a day or 2 days. The membranes that enclose the pellet's contents slow its dissolution, holding the organic material inside in a compact package. The density, achieved by the compression of organic matter through digestion, gives the pellet gravitational authority that the softer aggregates lack. A salp is a different kind of animal altogether. Salps are gelatinous, barrel-shaped, transparent, and they filter enormous volumes of seawater through their bodies to extract phytoplankton, sometimes forming chains of individuals, dozens of meters long, that pulse through the surface layer in a loose coordinated rhythm. They are voracious, and their fecal pellets are correspondingly large. A single salp produces a pellet that is many times denser than any loose phytoplankton aggregate of similar size, and it descends through the water column in something close to a straight line, while the softer particles around it wander and drift. Watching a salp pellet fall through water beside a loose aggregate of cellular debris is something like watching a stone fall beside a feather. The same destination, achieved at vastly different speeds by vastly different routes. This difference in sinking speed matters enormously to what survives its descent. An aggregate that takes months to reach the deep ocean passes through hundreds of meters of water inhabited by bacteria that are actively dissolving it, by small animals that eat it as it passes, by currents that disperse it sideways and break it apart. An aggregate that falls quickly bypasses much of this gauntlet. The fastest sinking particles carry more of what they contain to greater depths and do so with less loss. The ocean's transport of material from surface to deep is not a single uniform process. It is a mixture, a simultaneous, continuous, varied fall of particles at every speed between extremely slow and merely slow, all of it heading in the same direction, all of it carrying carbon downward, each component doing so at its own pace by its own physical logic. The size of marine snow particles varies across a range that is wider than it might seem. The conventional lower boundary of what counts as marine snow, the minimum size at which a particle is considered part of the system rather than merely a suspended fragment, is around 0.5 millimeter, which is roughly the size of a grain of fine sand. At that scale, a particle is barely visible, just at the edge of what a careful eye in clear water might detect as something rather than nothing. At the other end of the range are aggregates several centimeters across, the diameter of a small coin or of an eye. These large flocks are the most spectacular members of the marine snow community, though they are also the rarest. They form when conditions are particularly favorable for aggregation, when a phytoplankton bloom is peaking, when gel production is high, when the water is calm enough that collisions between particles produce adhesion rather than disruption. Under those conditions, an aggregate can grow over hours into something large enough to see clearly from a meter away, a loose, pale, rotating structure that descends with the imperious slowness of something that has all the time in the world, because it does. Inside a large marine snow aggregate, seen in the beam of a very bright light, the internal structure is surprising. It is not solid. It is more like a three-dimensional net, a sparse framework of gel threads and particle fragments with large empty spaces between them, the whole thing more void than matter, held in shape by the same adhesive properties that caused it to form in the first place. At its center, sometimes, there is something that was once a diatom chain, a string of phytoplankton cells linked end to end as they divided, each one surrounded by its silica shell, the whole chain now embedded in the aggregate's interior, like an insect preserved in resin, its geometric precision still intact, a record of an organism that lived and divided in the sunlit surface weeks or months before. Around this nucleus, layer after layer of fragments and gel have accreted, the way a pearl grows, not directed, not purposeful, simply the outcome of physical properties operating without any oversight. Bacteria do not wait for the aggregate to arrive at depth before beginning their work. By the time a marine snow particle has descended just a few meters from where it formed, it is already covered on its outer surface by bacterial cells. These bacteria are attached, not free-floating. They have adhered to the aggregate and are actively releasing enzymes that break down the complex organic molecules of the aggregate's outer layer into smaller, simpler molecules the bacteria can absorb. The process is slow relative to the rates of surface production, but it is continuous, and it means that every marine snow particle is being simultaneously transported downward and consumed from the outside in. A particle that falls for a long time loses mass, its edges become less distinct. The loose outer structure dissolves before the more protected interior. What arrives at depth, if anything does, is a reduced version of what started from the surface, compacted, partly transformed, its original composition substantially altered by weeks of bacterial attention. The water column through which marine snow falls is not a passive medium. It has layers, each with its own temperature and its own chemistry, and the passage from one layer to the next can change what a sinking particle looks like and what it carries. Near the surface, the water is warm and well mixed, and particles aggregate freely in conditions that favor gel formation. Below the warm surface layer, the temperature drops sharply across a boundary called the thermocline, a zone where descent through a few tens of meters can mean encountering water that is several degrees colder, denser, and moving at different speeds than the layer above. Particles crossing this boundary encounter different bacterial communities, different pressure, different chemical gradients. They do not stop. The aggregates are loose enough and buoyant enough in their own limited way to cross this boundary without shattering, and they continue downward into the water that lies below, darker and colder and slower. The first few hundred meters of the descent are the most eventful. This is where the water is most biologically active, where the most animals live, where grazing and fragmentation and dissolution are most intense. A marine snow particle descending through this zone may be intercepted by a small zooplankton animal and eaten, in which case its carbon continues the journey in a different form inside a living gut on its way to becoming a fecal pellet and sinking again, or being respired and released as dissolved gas. A particle may be broken apart by the feeding motion of a swimming animal and its fragments dispersed sideways in the water, where they may aggregate again with other fragments into a new particle, or where they may be small enough to remain suspended, drifting for days or months before settling again. Or a particle may pass through this zone relatively intact, carried downward by its own density, shrinking as bacteria consume it, but surviving. A phytoplankton bloom, seen from just below its own lower boundary, is a remarkable thing. From above the surface, a bloom is visible as a color change, a green stain spreading across the blue ocean over days and weeks. From below, from inside the water column, just beneath where the cells are concentrated, the view is different. The water is dense with material. Light enters from above, through a layer that is almost thick with life, filtering down through billions of tiny cells, each one deflecting and absorbing some fraction of it, so that what arrives in the water just below the bloom is fragmented, shifting, full of green-gold color that is the light the cells have not yet absorbed. In this water, just below the bloom's richest layer, the aggregation is happening in real time. Gel threads extend through the water between cells and cell fragments. Contacts are being made. Small particles are becoming larger particles. The first aggregates to form are already beginning to sink, carrying the first of the bloom's production downward toward the dark water below, before the bloom itself has even reached its peak. This is how the surface world enters the deep. Not in a single event, not through a sudden transfer, but continuously through the patient accumulation of small events, a cell dying, a fragment binding to another fragment, a pellet forming inside a small animal and sinking faster than anything around it, a gel thread extending across the space between two particles that would otherwise never meet, all of it adding up day by day, without pause, to a continuous slow rain of organic matter falling from the world of light into the world of permanent dark below. The scale of marine snow particles means that none of them individually carries much. A single aggregate a centimeter across contains a vanishingly small amount of carbon by any measure that matters to the atmosphere or the climate. But there are, at any given moment, an almost impossible number of them. Across the global ocean, across hundreds of millions of square miles of surface, particles are forming and falling continuously. The slow fall of one particle is multiplied by billions of simultaneous falls in the North Atlantic and the Southern Ocean, in the warm waters of the tropics, in the cold productive seas near the poles, all at once, always, without rest. The ocean is not simply a body of water. It is, among many other things, a machine for slowly moving the surface world into the deep. A fecal pellet produced by a large euphoacede, a krill-like animal a few centimeters long, the kind that forms massive schools in polar seas and is the primary food of whales, is a dense, dark, cylindrical object, roughly the size of a grain of rice. It is bound by a thin membrane that the animal's gut has deposited around the compacted contents. Inside are the fragmented, partially digested remains of the phytoplankton the animal has been grazing on for hours, cells broken down but not fully dissolved, their organic content still largely intact, packaged now into a form dense enough to sink at several hundred meters per day. Released into the water column from an animal feeding at the surface, this pellet begins its descent immediately, leaving behind the sunlit water where it formed and entering the progressively darker, colder water below. Within hours, it has traveled further than a loose phytoplankton aggregate would travel in a week. Within days, it has crossed the depth boundary where light gives out entirely and entered water that has not been touched by sunlight in any direct way. It continues downward. It is still being colonized by bacteria on its outer surface, still losing mass at its edges, but its dense interior is protected by the membrane, and most of what it carries will survive the journey intact, delivered efficiently to a depth that loose aggregates rarely reach before being entirely consumed. The variety of particles that constitute marine snow is in a sense a record of everything that has been living in the surface ocean above. The frustules of diatoms, those silica shells with their extraordinary geometric patterns of pores and ribs and radiating spines, different for every species, as precise and varied as snowflakes are in a different comparison, are among the most durable components of marine snow, outlasting the organic material of the cells they once enclosed. A diatom frustule can survive the descent through the water column and arrive on the seafloor still structurally intact, still legible as the shell of a specific organism, still carrying in its proportions information about the conditions under which the cell that built it once lived. Mineral fragments, clay particles carried by rivers to the ocean, wind-blown dust settling on the surface, volcanic ash from an eruption somewhere on land, join the organic material in the aggregates, carried downward in the same slow snow, sinking because they are denser than water and because they have found adhesion in gel threads to particles that give them structure and surface area. The snow is not clean. It is not simple. It is the mixed outfall of an entire living surface world caught by chemistry and slow gravity and sent downward into the dark. There is a particular quality to the movement of a large marine snow aggregate through still water. It does not fall the way a stone falls, not in a straight line, not accelerating. It falls the way a feather falls in a room with no wind, in a slow drift, occasionally deflected sideways by the slightest irregularity in the water it is passing through, occasionally rotating, occasionally stalling, and then beginning again. The water it descends through is not perfectly still. Even deep in the ocean, far below the reach of surface winds and waves, There are slow horizontal currents, internal waves, the gentle circulation driven by differences in temperature and salinity between water masses. A marine snow aggregate descending through this water does not travel straight down. It wanders slightly. It may be carried sideways by a current and then descend again when the current weakens. The particle that formed at the surface directly above a particular point on the seafloor may arrive, weeks or months later, at a point many miles away. The seafloor receives what the surface produced, but not necessarily directly below where the production happened. The relationship is diffuse. The delivery is spread. Watching from inside a submersible with bright exterior lights, the water around a marine snow aggregate during the aggregate's first hours of descent, it is possible to see the bacteria already at work on its surface. Not individually, the bacteria are far too small for that, but collectively, the bacterial film on the surface of an aggregate gives the edges a slightly hazy quality, a faint lack of sharpness where the dissolution is proceeding, where complex organic molecules are being broken into simpler ones and released into the surrounding water as dissolved carbon, returning to the water column rather than continuing the descent. The aggregate is doing two things simultaneously, sinking and shrinking. It is a vessel with a leak, moving toward a destination it may never reach in its original form, delivering its cargo in installments as it goes. Some marine snow particles do not survive the descent. They dissolve entirely, or are eaten entirely, or are broken apart by turbulence, or by the feeding currents of animals, and their constituent materials return to the water in dissolved form. The water column is full of this dissolved organic matter, a vast reservoir of carbon that was once living tissue and is now adrift in the ocean, neither sinking nor rising, available to bacteria that can absorb dissolved molecules directly. Much of what is produced at the surface enters this dissolved pool before it ever becomes part of a sinking particle, and much of what starts as a sinking particle dissolves back into this pool as it descends. The fraction that survives, that maintains its particulate structure through the full depth of the water column and arrives intact on the seafloor, is small, but it is not zero. Some particles make it, some always make it. The ocean is patient, and the descent is long, and a small but steady fraction of what begins at the top completes the journey to the bottom. Consider a marine snow aggregate that has just formed. It is a few millimeters across, large enough to count as marine snow by the standard measure, small enough to be nearly invisible in open water. It is pale, loosely structured, and it holds within it the fragments of perhaps a dozen phytoplankton cells, 3 or 4 mineral grains, a fecal pellet fragment from a copod that was feeding in the bloom, and an invisible covering of bacterial cells on every exposed surface. It began forming a few hours ago in the warm upper water where the phytoplankton bloom is still active and gel threads are abundant. It is now beginning its descent. Around it, in the same water, are thousands of similar particles in various stages of formation and decay, some larger, some smaller, some fresher, some already visibly dissolved at their edges. The water is full of these slow, pale, quietly falling things. Below, the blue deepens toward gray. Below that, the gray deepens toward black. The seafloor, where this particle is theoretically headed, is perhaps 3 or 4 miles down, a distance that, at the pace this particle is falling, represents a journey measured in weeks or months, not hours or days. The particle drifts downward. It rotates slightly. A current nudges it sideways, and it drifts, for a moment, almost horizontally, before gravity reasserts its patient claim, and the descent continues. This is the beginning of marine snow's story, not its implications, not its consequences, not the larger processes it is part of, or the communities it sustains, or the climates it shapes. Only this, a pale, loosely structured, slowly rotating aggregate, a few millimeters across, descending through dark water at the pace of settling dust, covering perhaps 5 meters over the course of a full day, heading downward through the blue and the gray and the black toward a seafloor. It will take weeks to reach, if it ever does. The ocean around it is vast and cold and almost entirely dark. The particle is very small. Its descent is very slow. And it began, as everything here begins, in the bright and busy world of light, at the surface, where a phytoplankton cell died at the right moment, in the right water, near enough to a gel thread to be caught by it, and the long, slow journey downward became possible. Somewhere below, in water the light has never touched, the fall continues. The aggregate drifts. It shrinks slightly as bacteria work at its edges, but its core is still intact, still holding the shape of cells that were alive a few weeks before, still carrying the carbon they fixed from the water and the air above the water, and the long chain of exchanges that connects a molecule of atmospheric gas to a living cell, to a dying cell, to a fragment, to an aggregate, to this slow, patient, turning particle. In the dark, it continues downward. There is no hurry in this. There has never been hurry in this. The ocean has been snowing slowly downward into itself for longer than there have been animals to observe it, and it will continue long after all of this has been forgotten, one pale and loosely structured particle at a time, descending toward a resting place it may take a season to reach. A single phytoplankton cell dies at the surface of the ocean in the pale light of a northern morning. It is smaller than the period at the end of this sentence. It has no weight a human hand could measure, and yet the moment its cell wall fails and its contents begin to disperse, something starts moving, something that will not stop for weeks or months, or in some forms for longer than that, a slow carbon-carrying descent through hundreds of meters of dark water toward a floor it may or may not reach intact. That one cell is not alone. It never is. In the surface layer of the ocean, where sunlight still reaches and photosynthesis is possible, phytoplankton exist in numbers that make counting feel beside the point. Billions of cells in a single liter of seawater during a bloom, each one fixing carbon dioxide from the water around it and converting it, using light, into organic matter. The ocean breathes in this way. It takes the carbon that the atmosphere has deposited into the surface water and folds it into living tissue, into the soft bodies of cells no eye can see without help, holding it briefly in biological form before releasing it again, downward. This is what oceanographers call the biological pump. The name suggests machinery, gears and pistons, something industrial. The thing itself is quieter than that. It is the sum of every cell that dies and falls, every pellet that sinks, every fish that descends at dawn, carrying the remnants of its night, feeding inside it. Across the entire global ocean, this process moves somewhere between 5 and 12 billion tons of carbon from the sunlit surface toward the deep each year. Not all of it arrives. Most of it does not. But the fraction that does has shaped the chemistry of the atmosphere across geological time, and the pump continues now, in this moment, in every ocean basin simultaneously, in the dark and in the light, in winter and summer, far from any shore. The journey begins at the surface, in the zone where light is strong enough to support photosynthesis. This sunlit layer reaches down perhaps 200 feet in the clearest open ocean water, shallower where the water is dense with particles, shallower still where storms have stirred the surface into opacity. In this layer, phytoplankton bloom and die and become the raw material of the pump. Their deaths are not violent or dramatic. A cell reaches the end of its division cycle and fails to divide again. Its membrane weakens. Its contents, the carbon-rich sugars and proteins and lipids built from months or hours of photosynthesis, begin to leak into the water around it, and the cell itself starts to aggregate with neighboring cells, with fragments, with the sticky exopolymer particles that hold the whole system together like an invisible glue. Bacteria arrive almost immediately. They are always nearby, always present in the surface layer, in numbers that dwarf even the phytoplankton they shadow, and their response to a dying or dead cell is swift and total. Enzymes are released from the bacterial cell surface into the surrounding water, enzymes that begin dismantling the organic molecules of the phytoplankton cell into their simplest dissolved components. Carbon dioxide forms, nutrients, nitrogen, phosphorus, dissolve back into the water. The carbon that the phytoplankton fixed from the atmosphere is returned to the water in dissolved inorganic form, available to be exchanged with the atmosphere again, its brief biological life over. This is the first and most complete interception of the pump's cargo. Most of the carbon produced at the surface never descends at all. It is recycled in the surface layer before it can aggregate and sink, turned back into the dissolved form from which it came. What escapes this first interception escapes because it becomes heavy enough to sink, or because it is packaged in a form that resists immediate bacterial breakdown. Aggregates form, marine snow, and within the aggregates, bacteria are already working, but the interior of a large, dense flock is harder to reach, and the descent begins before the bacteria can fully dismantle it. Zooplankton fecal pellets escape because their dense membrane-bound packaging slows the exposure of their contents to enzymatic attack. The material that falls is the material that outruns its decomposers, at least for a while. Below the sunlit layer, the water changes. The light does not go out at once. It fades, blue deepening through shades that have no common names, the wavelengths failing one by one until only the shortest remain, a faint, cold blue that is more darkness than light. At 200 feet, a human eye can still detect the sun as a diffuse brightness above. At 1,000 feet, the last traces are gone, and it is in this transition zone, this slow fade from pale blue to absolute black over the span of several hundred feet, that the ocean's most intense biological interception takes place. This middle region of the water column, extending from roughly 600 feet to just over 3,000 feet below the surface, is called the mesopelagic zone. It has other names. The twilight zone, because of the quality of light at its upper margins, that faint and failing blue that gives way to nothing. The midwater, because it is neither surface nor floor, but the vast, unconsidered space between. Whatever name it carries, this is the place where the biological pump loses most of its cargo, and the losses are enormous. Somewhere between 70 and 90% of the carbon that sinks out of the sunlit layer is intercepted, processed, and returned to dissolved form within the mesopelagic zone before it descends further. The pump is porous at every level, and this is the level where the porosity is greatest. Bacteria carry most of the responsibility for that interception. A marine snow particle descending through the mesopelagic is not traveling alone. Its surfaces are densely colonized, more densely than the water it passes through, because the particle is an island of concentrated organic matter in a desert of dilute, dissolved substances, and bacteria that find it have for a time everything they need. The same enzymatic process that began at the surface continues here, only more slowly because the water is colder and the pressures are higher. The particle shrinks as it falls. Its loosely aggregated outer layers dissolve into the surrounding water. The bacteria consuming it excrete carbon dioxide and nutrients that drift into the column behind the particle's path, a faint chemical wake that disperses into the surrounding dark. What began at the surface as a centimeter-wide flock may arrive at 3,000 feet as a fragment half its original size, and it will have lost most of its carbon content to the water column it traveled through. The mesopelagic zone has a second, stranger mechanism of interception, one that operates not through passive decomposition, but through active biological transport. Every day, across every ocean, one of the largest organized animal movements on the planet takes place. At dusk, when the surface darkens, vast numbers of small animals begin to rise. Copepods the size of a grain of rice, small fish the length of a finger, shrimp, krill, bristle-mouthed fish with rows of tiny light organs along their bellies, all of them ascending through hundreds of feet of water in the space of an hour or two, rising toward the surface to feed in the dark on the phytoplankton and smaller zooplankton there. Then, at dawn, they descend. The light strengthens above them, and with it the predators that hunt by sight, and the vast cloud of migrating animals sinks back down through the water column to the depths where darkness protects them through the daylight hours. They carry with them whatever they have eaten at the surface, carbon fixed by phytoplankton, packaged now inside their bodies in fats and proteins. And at depth, over the course of the day, they breathe and excrete and defecate. They release that surface carbon into intermediate and deep water in forms dissolved or particulate, injecting it hundreds of feet below the level where it was first fixed. This is called active flux. It is the biological pump, operating not through the passive sinking of particles, but through the intentional daily migration of animals that feed high and metabolize low. The animals themselves do not know they are transporting carbon. They are following light and following food, rising to abundance and descending to safety, doing this each day of their lives as their parents did and as their offspring will. The carbon is a consequence of the feeding, not a purpose. But the consequence is real and measurable. Active flux from diel vertical migration adds a significant portion to the carbon that the pump carries downward, a contribution that in some ocean regions rivals the passive sinking flux of marine snow itself. Below the mesopelagic zone, the interception continues but slows. The water in the bathypelagic zone, below 3,000 feet, down to perhaps 13,000, is cold and almost perfectly dark, and the community of organisms there is smaller, sparser, and more patient. Particles that reach this depth have already been processed repeatedly. They are less nutritious than they were at the surface. They are smaller. They sink more slowly because they have lost their densest, most compact components to bacterial remineralization. But they are still falling, still carrying carbon, and they are still being intercepted, still being consumed by bacteria and by the sparse midwater animals that pick them from the column. A single descending particle in the deep water is surrounded by cold on all sides. The water at these depths holds temperatures just a few degrees above freezing, sometimes less in the polar basins, and in the cold the biochemical processes of decomposition proceed slowly enough that some organic matter survives for a long time. Pressure here is immense, 100 atmospheres and more, then 200, then 3, pressing on every surface with a force that has reshaped the chemistry of the organisms that live here, changed the form of their enzymes and the structure of their cell membranes into versions that function under compression rather than failing under it. Through all of this, the particle falls. It falls slowly enough that a single marine snow aggregate might take weeks or months to cross the mesopelagic zone and reach the deeper water below. It falls in a world that from the outside looks featureless and homogeneous, dark water, cold, still, but is in reality a continuous vertical gradient of changing temperature, changing pressure, changing oxygen concentration, changing biological community, changing rates of decomposition. Each 100 feet downward is a different environment, with different inhabitants, different rates of chemical processing, different fates waiting for the carbon the particle carries. And the particle is not falling straight. Currents at all depths move horizontally, and a particle descending through them is carried sideways as it descends, tracing not a straight line, but a long, slow diagonal through the water column. The place where it arrives, if it arrives, may be miles from the place below which it was produced at the surface. The sea floor is not receiving a direct accounting of what the surface above it produces. It is receiving a shifted, averaged time-delayed signal, the product of surface productivity from a broad area transported laterally as it descends. What survives all of this, what carries through the mesopelagic and the bathypelagic and arrives finally in the deep ocean, the true deep, the abyss, the waters below 13,000 feet, is a fraction so small it might seem to suggest the pump has failed. 1 to 3% of the total carbon fixed by phytoplankton in the surface layer reaches the deep sea floor. The rest has been remineralized, returned to dissolved inorganic form, recirculated through the water column by migrating animals, or recycled in the surface layer before it ever began to descend. Only 1 or 2 carbon atoms out of every 100 fixed at the surface complete the full journey. There is a temptation to hear that number as a failure. It is not. The ocean is vast in a way that resists casual statement. The surface layer alone covers most of the planet, and the photosynthesis occurring there is measured in billions of tons per year. 1% of billions of tons is still tens of millions of tons. Across geological time, across hundreds of millions of years of ocean biology, those millions of tons per year have accumulated in deep seafloor sediments into deposits of organic carbon that dwarf anything on land. The pump is not efficient in the way a machine is efficient. It is efficient in the way a vast and ancient process is efficient, by operating at a scale where even small fractions are immense, and by operating without pause across all of geological time. We'll inquire the dissolved inorganic carbon that the pump returns to the water column during remineralization does not immediately return to the atmosphere. Deep ocean water is isolated from the surface by the density stratification of the ocean. Warm, light water floating above cold, dense water with no easy mixing between them. Carbon dissolved at depth stays at depth, removed from atmospheric exchange for decades, centuries, or longer, depending on the circulation of the deep ocean. When a particle is remineralized in the mesopelagic zone, and the carbon dioxide it releases dissolves into the surrounding cold water, that carbon dioxide is, for the moment, sequestered, held in deep water at a pressure and temperature that keeps it dissolved rather than returning it to the air. The ocean holds it there, patient and dark, until the slow overturning of the deep circulation eventually brings that water back to the surface and allows the exchange to begin again. The time scales involved span centuries. Water that descends into the deep North Atlantic carries its dissolved carbon load for 1,000 years or more before it resurfaces in the North Pacific as part of the ocean's great conveyor circulation. Carbon, that was remineralized from a sinking particle off the coast of Iceland in the medieval period may still be circulating in deep water today, its return to the atmosphere still centuries away. The pump does not simply move carbon from the surface to the floor, it moves carbon from one timescale to another, from the fast timescale of photosynthesis and bacterial decomposition to the slow timescale of ocean circulation and geology. In the mesopelagic zone at midday, the darkness is not peaceful in the way the darkness of a sealed room is peaceful. It is populated, in motion, full of the small metabolisms of 1,000 organisms carrying out their separate lives in the cold and pressure. A bristle-mouthed fish, silver and barely visible, hovers motionless in the water, waiting. Bacteria, too small to see without magnification, cover the surface of every passing particle. A copod feeds on a fragment of marine snow, breaking it with small, precise mouthparts, extracting what nutrition remains, excreting a new pellet that will itself sink and be consumed by something else. All of this takes place in silence that is not quite complete. Sound travels well in water, better than in air, and the deep ocean is not silent. It carries the low-frequency sounds of whales many miles away, the distant creak and groan of tectonic stress, the broadband hiss of distant storms at the surface. But in the mid-water, where there are no breaking waves, no wind, no human machinery, the soundscape is the quietest available on the planet's surface, a deep ambient background against which the biological sounds of feeding and movement are almost inaudible. The particles continue to fall through all of it. Some are large enough to carry bacteria from the surface layer down into the mesopelagic, acting as transport vehicles for microorganisms that would not otherwise descend. Some are colonized by new bacteria at each depth, different species adapted to the temperatures and pressures they encounter. The particle's bacterial community turning over completely between the surface and the deep. The particle itself changes, shrinks, darkens slightly as the more labile organic molecules are consumed, and the more resistant compounds remain, becomes denser in some ways and more porous in others, its structure shifting over weeks of descent. A fecal pellet produced by a small copod at the surface has a different fate than the loose, fluffy aggregate it might have settled beside. Dense and membrane-bound, the pellet sinks faster, not the 1 to 10 meters per day of a slow aggregate, but hundreds of meters per day in some cases. At that speed, it passes through the mesopelagic zone in a matter of days rather than weeks, giving the bacterial community less time to process it, giving the migrating zooplankton less opportunity to intercept it. Dense, fast pellets are among the most efficient vectors of carbon transport to depth. They carry more carbon per unit of surface area exposed to bacterial attack, and they carry it down faster than the bacteria can consume it. Not all fecal pellets are equal. The pellets of large zooplankton, salps, euphausiids, large copods, are denser and larger than those of smaller animals, and they sink correspondingly faster, transporting their carbon load to depth more efficiently. Salps in particular produce pellets that are among the fastest sinking objects in the open ocean, compact, dense cylinders that descend with a near directness that contrasts sharply with the wandering drift of soft marine snow aggregates. When salps bloom, and they bloom in enormous numbers in some ocean regions, the seafloor beneath them receives a rapid, intense pulse of organic carbon, a brief flooding of material after weeks or months of relative scarcity. The pump is not constant. It pulses with the rhythms of the surface, the annual cycle of blooms, the interannual variations of climate, the episodic events of storm and cell bloom, and krill swarm that produce sudden surges in the export of organic carbon. The mesopelagic community responds to these pulses in ways that are still being worked out, the consumption of carbon accelerating when supply increases, the community expanding and contracting in ways that modulate how much carbon passes through and how much is returned to dissolved form within the zone itself. Below the mesopelagic zone, as depth increases and temperature drops and pressure mounts, the organisms that intercept sinking particles become fewer. In the bathypelagic zone, the water is effectively lightless in a way that the mesopelagic is not quite. No photons of any wavelength reach here from the surface, and the only light comes from bioluminescence, from the organs of fish and shrimp and squid that produce their own cold blue-green glow. Particles that sink through the bathypelagic are in a different world than they were above the mesopelagic, a world where the interception rate is lower, where decomposition continues, but at a slower pace, where the carbon they carry has a better chance of reaching the deep. And deep means something specific here, below roughly 13,000 feet, in the cold, near-freezing water that covers the greatest extent of the ocean floor. The particles that arrive are the survivors, not of any competition or struggle, but simply of the slow attrition of remineralization and consumption that has claimed most of what was produced at the surface. They are fewer, they are smaller, they are less nutritious, but they are real, and they arrive, and the communities of the deep sea floor have organized themselves around their arrival across hundreds of millions of years of evolutionary time. 1 or 2 carbon atoms in every 100. That is what completes the descent. Held in a flock, or pellet, or fragment, carried through 2,000 meters of water in the darkness, past every organism that could intercept it, arriving finally in the cold and the pressure and the silence of the deep ocean, where the water has not seen the surface for 1,000 years or more. The carbon arrives, changed from what it was. The sugars and proteins are largely gone, consumed by bacteria at every level of the descent, and what remains is the more resistant fraction, the complex organic compounds that have resisted enzymatic attack, the mineral-associated organic matter that is protected by its attachment to clay particles and sediment grains. What arrives in the deep is not what was fixed at the surface. It is a distillation, the fraction of surface productivity that survived the whole long passage through an ocean that was trying to recapture it at every depth, every moment, from the surface bloom to the final meters before the floor. It is changed and diminished and small, and it is enough. The deep ocean holds it. The cold water dissolves what dissolves and carries the rest to the floor, and the floor receives it, receives the 1 or 2 atoms out of every 100, decade after decade, bloom after bloom, spring after spring at the surface, while the deep remains dark and cold and patient, accumulating the slow carbon delivery from above into the sediment that has been building there since the first phytoplankton evolved and died and fell hundreds of millions of years ago. What the pump does is not dramatic. There are no sudden transformations, no visible reactions, no moment at which the carbon crosses a threshold and becomes permanently buried. It is a process of gradual removal, a slow, continuous subtraction of carbon from the atmosphere-ocean surface system, and a slow, continuous addition to the deep ocean and the seafloor below it. The drama, if there is any, belongs to geological time, to the way this process, operating across millions of years, has shaped the composition of the atmosphere and the temperature of the planet, holding carbon in the deep ocean rather than the air, maintaining the conditions that have allowed complex life to persist and diversify. The pump is not separate from the life that surrounds it. It is made of that life. Every copod that feeds and defecates, every bacterial cell that attaches to a sinking aggregate, every fish that descends at dawn and releases its surface-fixed carbon into the dark at depth, all of them are the pump. It has no structure apart from their bodies and their metabolisms and their daily movements. It is the most distributed biological process on the planet, operating in every ocean basin, at every depth, with no center and no periphery, continuous across the widest surface of the Earth. A particle falls now, somewhere in the North Atlantic, through cold blue water in the failing light below the surface bloom. It is small, a few tenths of a millimeter, a loose aggregate of diatom fragments and bacterial cells and the sticky polysaccharide threads that hold them together. It sinks slowly, perhaps a few meters per day, and the bacteria on its surface are already at work, already dissolving the outer edges of the aggregate into dissolved carbon dioxide and nitrate that drift away behind it. It will lose most of what it carries before it reaches the bottom. It will probably not reach the bottom at all, but it is moving, and as it moves, it is carrying with it a small fraction of this spring's surface productivity toward whatever depth it can reach. One small delivery in the billions of deliveries that together keep the pump working, and the deep ocean fed, and the atmosphere in its long, slow balance with the sea. The particle falls in the dark, the bacteria work, the cold water holds everything in suspension. The particle, the bacteria, the dissolved carbon dioxide they produce, the faint wake of their collective chemistry trailing upward into the water. The particle has already left behind. From above, the surface of the ocean is bright and green, with the blooms still underway. From below, the light is a pale diffuse glow that fades with distance, and the particle descends through the fading, through the gradient, through the long passage from light to dark, carrying one or two carbon atoms that may, over the weeks and months that follow, travel the full depth and arrive in the cold and silence of the abyss, where they will rest, dissolved or settled, removed from the atmosphere for centuries and perhaps longer. The surface bloom will fade. The particles will keep falling long after the bloom has ended, because the slow aggregates take weeks to descend, even a few hundred meters, and the pellets take days, and the midwater migrants descend each morning with their cargo, and the biological pump does not pause when the bloom ends, but continues at its reduced pace through summer and autumn and winter until the next bloom begins. The pump does not rest. It slows and quickens and slows again, but the falling never stops. The bacterial consumption never stops. The deep ocean never stops receiving the one or two atoms in every 100 that complete the descent. The particle falls. The ocean holds it. And somewhere far below, in water that has been cold and dark for longer than any record of human events, the smallest fraction of a spring morning's photosynthesis arrives at last, and the deep receives it, and everything continues. Standing on the floor of the deep ocean, in water so cold it holds just above freezing, in darkness so complete that no shadow has ever formed here, the eye adjusts and finds nothing to adjust to. There is no light. There has never been light at this depth. The pressure at 4 miles down is enough to compress the air in a human lung to the size of a fist. The silence is not empty. It carries the faint, low, almost geological groaning of the water column above, the slow mechanical sound of an ocean weighing down on the rock beneath. And yet, when the eye is given instruments it does not naturally possess, when the darkness is replaced by a sensitivity borrowed from technology, the first thing visible is not the darkness, it is the snow. From kilometers above, a slow rain descends. Individual particles drift with the unhurried patience of dust settling through still air in a room where no one has moved for hours. They are pale, they are loosely structured. Against the absolute black of the deep water, they are the only things that can be called, in any sense, bright. They fall too slowly to seem to be falling. They turn as they descend, rotating without apparent cause, and the occasional one catches what little there is to catch, a faint bioluminescent flash from some passing body, perhaps, or simply the scattering properties of their own fibrous surface, and for a moment they are almost visible to a hypothetical eye that has learned to look. Then the moment passes, the particle continues downward, others follow behind it. This is the world that the deep sea floor's inhabitants have organized their entire lives around receiving. A sea cucumber moves across the sediment at a pace that makes the falling particles above it seem fast. Its body is soft, elongated, pale as a root, grown in a lightless cellar, and it draws the sediment beneath it into one end and passes it through steadily, extracting from it whatever organic matter remains. The sediment on the abyssal plain is old, most of it is mineral, but threaded through the mineral, pressed into the spaces between grains, aged and chemically altered past easy recognition, is the carbon that arrived from above over thousands of years. The sea cucumber does not hurry. Hurrying in this environment serves no function. The food is not concentrated in a single place or available in a sudden flood. It is everywhere, thinly distributed, requiring patience rather than pursuit. And the sea cucumber has patience in whatever form patience takes, in a body that has no brain to grow impatient. It moves on a nearly imperceptible course, tracing a slight arc across the sediment surface, and the path behind it is just distinguishable from the undisturbed sediment around it. The material it has passed through is marginally finer, slightly more compacted, altered by passage through a body that extracts and returns with different chemistry. Deep-sea holothurians, which is the name given to sea cucumbers in their formal taxonomy, are among the most abundant animals on the abyssal plain in terms of total biological mass. In some regions, they cover the seafloor in numbers that, seen from above with proper instrumentation, give the impression of a living surface. They move when food becomes locally depleted, following gradients so subtle that the chemistry of the water 100 feet away must carry the signal. It is navigation without any instrument the human mind can easily picture, but it is accurate enough to have sustained these animals through geological spans of time that encompass the entire history of complex life many times over. The sediment the sea cucumber passes through holds, in its older, deeper layers, a record of every year the surface ocean has produced a bloom, since this patch of abyssal floor was last disturbed by currents or geological activity. The top layer is palest, most recently arrived, holding the faint green-gray tint of phytoplankton pigments, not yet fully broken down. Below it, darker and more compact, is the material of previous years and decades. Below that, in layers that require instruments to distinguish from one another, the record extends backward in time, centuries, millennia, longer. The sea cucumber ingests from the top, from the freshest material, drawn there by the slightly elevated organic content of recent arrivals. It does not know it is reading history. It is simply eating, a few meters away, where the sediment gives way to a slightly harder substrate. A different kind of creature waits with its arms extended into the slow movement of water just above the floor. Brittle stars belong to the same broad group as sea stars, sharing the five-fold symmetry and the general architecture of radially arranged arms extending from a central disk. But where sea stars move across surfaces on tube feet and pry open prey, brittle stars hold their arms aloft, each one reaching into the slight bottom current with a delicacy that makes the word fishing seem too mechanical. Along each arm, hundreds of tube feet extend like fine hairs, each one capable of capturing a particle that drifts close enough to touch. In regions where carbon flux from above is steady and sufficient, brittle stars aggregate in densities that cover the seafloor completely, dozens of individuals per square meter, arm touching arm, each one harvesting a slightly different current band. The living carpet they form is not organized or cooperative in any social sense. Each animal acts independently of the others, but the effect, seen at any scale, is of a coordinated filter stretched across the bottom of the ocean, intercepting marine snow in its final centimeters of descent. A particle that has spent weeks falling from the surface layer enters the zone of the brittle star bed. It has already lost most of its original mass. Bacteria have worked on it throughout the descent, dissolving its more accessible organic components into the surrounding water. What remains is a denser fraction, mineral-bound material, tightly packaged organic compounds resistant to easy enzymatic breakdown, fragments of structures that were once diatom shells or the membranes of fecal pellets. The particle passes through the brittle star's current zone. A tube foot touches it. The foot bends, drawing the particle toward the arm. Another foot contacts it. Within seconds, the particle has been passed in a chain of small mechanical grips toward the central disk, where the brittle star's mouth, a five-pointed structure on the underside, receives it. The whole transaction takes less time than the particle spent in its final hour of descent. What required weeks to arrive is consumed in moments. The deep ocean floor is not uniformly covered by brittle stars. Their density follows the availability of food from above, and the availability of food from above follows the productivity of the surface layer, which in turn follows the nutrients available in the water that the surface layer draws on. In the open ocean far from continents and upwelling zones, where the surface water is warm and stratified and nutrient poor, very little marine snow forms, and the seafloor below receives only the thinnest continuous trickle of particles. In these regions, the brittle star beds thin to scattered individuals, and the sea cucumbers between them travel greater distances between patches of worthwhile sediment. The abyssal plain in these low-flux regions is among the most food-limited environments on Earth, an enormous expanse of cold, still, dark, featureless water and sediment, where the slow business of existence requires extraordinary efficiency. Closer to productive surface waters, below the seasonal blooms of temperate and subpolar regions, and below the upwelling zones where cold, nutrient-rich water rises from depth, the floor beneath is correspondingly richer, and the organisms that live there are proportionally more numerous and active. The chemical texture of the sediment changes across these gradients. In high-flux regions, the sediment holds measurably more organic matter, more nitrogen, more phosphorus, more of the elements that biological processes require. In low-flux regions, the sediment is more purely mineral, more aged, more thoroughly processed by the organisms that have worked through it over time. A handful of sediment from a high-flux region would look and smell different from a handful taken from the center of a subtropical gyre, one faintly biological, slightly soft, the other dry and mineral and inert as stone. The organisms living in each know the difference, though not in any way that involves knowing. Among the smallest of these organisms, too small to see and present in numbers that resist easy imagination, are the bacteria. They are in the sediment. They are on the surfaces of every particle that has arrived. They are in the water column immediately above the sediment, held there by density gradients and chemical cues that keep them concentrated at the boundary between water and sediment. These deep-sea bacteria are not the same as bacteria that live in warm, sunlit, well-oxygenated waters near the surface. The pressure at the abyssal seafloor would crush most surface bacteria entirely, rupturing their cell membranes and disrupting the molecular machinery their chemistry depends on. The bacteria at 4 miles down are pressure adapted. Their cell membranes are built with fatty acids that remain fluid under conditions that would solidify a surface bacterium's membrane into something solid and non-functional. Their enzymes are shaped differently at the molecular scale, designed to fold and function correctly at temperatures near freezing and pressures that would stop most biochemical processes entirely. They are called piezophiles from the Greek word for pressure, and they are the final agents of decomposition in the deepest parts of the ocean. A piezophile cell, resting on a sediment grain in water just above freezing, secretes enzymes into the surrounding water. The enzymes are proteins folded into shapes that fit the surfaces of specific organic molecules, the way a key fits a lock, and under pressure, those shapes hold. The enzyme contacts a fragment of an ancient fecal pellet, a molecule of complex carbohydrate that has survived the entire journey from the surface and has not yet been broken down by any of the bacterial communities it passed through on the way. The enzyme cleaves the molecule. The fragments are small enough to be taken up directly by the bacterial cell through its membrane. Inside the cell, those fragments fuel the chemistry of metabolism, powering reproduction, maintenance, and the continued secretion of more enzymes. The cell divides. Slowly, another cell begins working on the same particle. The process has no urgency. At these temperatures and pressures, metabolic rates are a fraction of what they are in warmer, shallower water. But the process is steady, and over time, over the long, cold, dark time that the deep ocean affords in abundance, it is thorough. The fecal pellet that a copod produced at the surface is after weeks of descent and months of bacterial processing on the seafloor, reduced to the mineral fraction it always contained, plus the most chemically stable organic molecules available, compounds so resistant to breakdown that they will remain in the sediment for centuries before being fully mineralized. The carbon in those compounds is, for practical purposes, removed from the ocean's active circulation. It is buried, not in the geological sense of compression and transformation into rock, not yet, but removed from the water column, removed from the potential for rapid return to the atmosphere, stored in a cold, dark, low-energy environment, where the processes that would break it down operate at the pace of geological rather than biological time, some distance from the brittle star bed, where the sediment surface is just slightly more uneven, a low rise of a few centimeters, barely perceptible, produced by some slow geological process, or by the accumulated activity of the burrowing organisms beneath. The sediment is patterned by holes and mounds. The holes are entrances to burrows. The mounds are exits, or the accumulated castings of animals that move through the sediment from below and deposit processed material at the surface. Polychaete worms are responsible for much of this topography. They are not a single species, but a vast and diverse group. The polychaetes include thousands of described species, ranging from thin, hair-like forms barely a centimeter long to large, muscular burrowers as thick as a finger and many centimeters in length. In deep-sea sediments, where the soft, fine-grained material extends to great depths below the sediment surface, polychaetes penetrate far into the sediment column, turning it over, redistributing organic matter that has settled on the surface down into the depths below and bringing older material from below back up. The process this creates, bioturbation, the biological mixing of sediment, has consequences that extend well beyond any individual worm. When a polycatid draws fresh phytodetritus down from the sediment surface into its burrow, it buries recently arrived organic matter before the surface bacteria have had time to fully mineralize it. The material arrives in deeper sediment layers, already partially processed, but still holding a significant fraction of its original organic content. There, in the lower oxygen conditions of deep sediment, decomposition slows further. The organic carbon spends more time buried and less time in contact with the oxygen and organisms that would return it to the water as dissolved inorganic carbon. The worm, in doing nothing more than eating and moving, acts as a pump that pushes organic carbon downward into the sediment and extends the time before it is returned to circulation. At the same time, the burrow itself is a channel, where water moves through it. By the worm's own activity, polychaetes ventilate their burrows, drawing oxygenated water from above the sediment down into the depths of the burrow to support their own respiration. This irrigation carries dissolved oxygen into sediment layers that would otherwise be entirely anoxic, changing the chemistry of those layers and allowing aerobic bacterial decomposition to occur at depths where it would otherwise be impossible. The worm is simultaneously burying carbon and creating the conditions for its eventual breakdown. Both effects are real. Both matter. The net result for any given patch of sediment depends on which effect dominates, and that balance differs across the range of species, depths, and sediment types found across the global deep ocean. A polychaete moves through its burrow with a slow rhythmic contraction of its body segments, its head end, where sensory structures cluster, probes the sediment ahead of it. The organic matter it detects is not the fresh, bright green phytoplankton of the surface bloom. That material arrived weeks ago and was immediately colonized by bacteria and consumed in part by the brittle stars and the surface-dwelling holothurians. What the polychaete encounters in the upper sediment is already processed, already darkened, already minerally bound, but it is still food. Within its mass among the mineral grains are fragments of resistant organic compounds, dead bacterial cells, and the structural remains of organisms that have been partially broken down but never fully remineralized. The polychaete extracts nutrition from this material with an efficiency that the surface ocean's brighter, faster food webs do not require. In a food-limited environment, thoroughness replaces speed. And then, sometimes with no warning, the sediment can register, and no preamble, the water column carries quickly enough to prepare. Something arrives that is different. The chemical signal reaches the seafloor first. It travels as a dissolved plume, carried by the slight, slow currents that move along the bottom, spreading outward from a point. It is sulfur, partly. It is amino acids and lipids. It is the chemical language of a large quantity of biological material at a stage of decomposition that speaks loudly to any organism with the chemical receptors to hear it. The signal moves outward through the dark water, and from distances that are, by the scale of the abyssal plain, not far at all, but which, by any human sense of distance, would be surprising, the first responders begin to arrive. Lysian acid amphipods are small crustaceans, rarely more than a few centimeters in length, that live a largely wandering existence on and just above the abyssal seafloor. They are pale, almost translucent, and built with the compact, multi-limbed architecture of crustaceans adapted to a three-dimensional world. They swim as readily as they walk, and they are capable of sustained movement through the water column above the sediment when a chemical signal is strong enough to justify the energy cost. When the signal reaches them, they turn, they orient to the gradient, toward higher concentrations of the chemical signal, and begin moving. They are followed by others. The signal is strong enough that it reaches individuals spread across an area hundreds of meters across. Each one turns and moves. None of them knows the others are also turning and moving. Each one follows the same gradient independently. The result is a convergence, thousands of small crustaceans moving through cold, dark water toward a single point on the seafloor. A whale carcass lies on the sediment. It is large, the skeleton of an animal whose living mass might have equaled a school bus in length and far exceeded one in weight. It arrived in stages. The soft tissues fell first, sloughing from the body during the long fall from the surface, some of it drifting away as it descended. The main carcass arrived more or less intact at the surface of the sediment, striking it with enough force to disturb the upper layers and send a cloud of fine particles billowing outward. That disturbance alone was an event, for the slow sediment, which accumulates at rates of millimeters per thousand years. The sudden deposition of a large organic mass was an input without parallel in any normal time frame. The surface area of the carcass, now in contact with the cold, still water of the deep, began immediately to release chemical signals. The signals moved outward, and the waiting, wandering organisms of the deep sea floor began to come. The first lysine acid amphipods arrive. They are already feeding before the density of their swarm has built to its eventual scale. More arrive. The population converges from every compass direction, from distances that took hours to cross. Within a day, the carcass is covered. The amphipods move over one another in a slow, dense mass, not frantic, despite the number of individuals, because the deep ocean cold renders even the fast metabolizing world of crustacean scavenging into something unhurried by surface standards. They are thorough. Soft tissue that the descent left intact is consumed. The blubber, rich in lipids, is taken. The organs, the muscle, the connective tissue, what they cannot easily process, they fragment and leave. What they fragment is colonized by bacteria. The bacteria work on the fragments. The amphipods work on the bacteria-covered fragments. At the same time, mobile scavengers from greater distances are still arriving. Hagfish move in long, slow curves above the sediment, following the chemical signal. Sleeper sharks, which range widely through the deep ocean at depths that would be extreme for most sharks, appear over the course of days. Each new arrival is another layer of consumption applied to what the earlier arrivals have left. The carcass diminishes. What was enormous by the scale of the abyssal plain, a sudden and tremendous concentration of organic matter, is reduced progressively. Bone emerges from the surrounding tissue as the mobile scavengers work through the softer material. The bones are dense, lipid-soaked, and will persist long after everything else is gone. What a single large carcass delivers to the seafloor in organic carbon is something that the continuous background rain of marine snow across that same area of seafloor could not deliver in hundreds of years. The organic input is concentrated in time and space. It is a pulse rather than a drizzle, and the community that responds to it does so in layers. The mobile scavengers first, then the enrichment opportunists, then, when the bones remain and the softer scavengers have moved on or died, the chemosynthetic community. The bones of a large whale decompose slowly in the cold, dark water. Bacteria colonize the bone surface, penetrating the porous mineral matrix, reaching the lipids stored inside. In the oxygen-limited interior of the bone, Anaerobic bacteria break down the stored lipids through a process that produces hydrogen sulfide as a byproduct. The hydrogen sulfide diffuses outward from the bone through the sediment and into the overlying water. And to hydrogen sulfide, certain bacteria have a very particular relationship. They can use it as an energy source, oxidizing it in a process that releases energy they use to fix carbon from dissolved carbon dioxide, building organic matter without any sunlight involved. They are the base of a food web that has no connection to photosynthesis whatsoever, and other organisms have evolved to harvest the organic matter these bacteria produce. Muscles colonize the bones. Small, specialized muscles, their tissues harboring communities of chemosynthetic bacteria within their own cells, extract energy from the hydrogen sulfide diffusing through the water around them. Clams settle into the sediment at the base of the bones and feed on the same sulfide-rich fluid. Polycha ate worms of specific species, not the generalist burrowers of the background sediment, but specialists that occur only at these sites, form dense mat-like aggregations on bone surfaces. Tiny snails graze the bacterial mats. Amphipods continue to pick at everything, omnivorous and persistent. The whole community, which does not exist anywhere else, which has assembled specifically here because of the whale, which will disperse or die when the whale's chemical bounty is finally exhausted, is an ecosystem within the ecosystem of the abyssal plain. It lasts decades. In cold, deep water, the bones of a large whale can fuel this community for 50 years or more before they are finally fully mineralized. The ordinary background rain of marine snow continues to fall throughout this entire sequence of events, indifferent to the drama of decomposition below. Particles descend past the whale fall, past the mussels and the polychaetes, and the swarms of amphipods, settling on the surrounding sediment exactly as they do everywhere else on the abyssal plain. The community clustered around the whale bones is an interruption in the monotony of the background system, an island of elevated energy within a sea of minimal flux. When it ends, when the bones are finally spent and the chemosynthetic organisms die back for lack of chemical fuel, the sediment will hold the mark of it, an elevated organic layer, a changed chemistry, a slightly different community of background bacteria for a long time after. The sediment remembers what has happened to it in the only way sediment can remember, by holding the chemical record of the past in its layers. The polychaetes that burrowed through the sediment around the whale fall have worked the enriched material down into the layers below the surface. The carbon that arrived in a single large pulse is distributed through the upper decimeters of sediment, mixed into the background, its distinctiveness gradually diluted by the slow accumulation of new material arriving from above. Over centuries, the carbon from that whale becomes indistinguishable from the carbon that arrived as marine snow in the ordinary course of things. It is all, in the end, part of the same system. Back on the undisturbed abyssal plain, the sea cucumber continues its path. It has moved perhaps a meter since the narrative began following it, an enormous distance by its standards, covered over hours. The sediment it moves across is, in this region, slightly enriched, not because of any whale fall or extraordinary event, but because the surface waters above are productive enough to sustain a continuous rain of marine snow at rates that allow the sediment to hold a modest but measurable organic content. The sea cucumber is not searching for anything in particular. It is not choosing its path based on a calculated assessment of the sediment's organic content. It simply moves, and as it moves, it feeds, and it feeds well enough to live and in time reproduce. The water above the sea cucumber is very cold and very still. At this depth, the currents that structure the surface ocean have been reduced to slow, broad drifts. Bottom water moving laterally at a pace of centimeters per hour rather than the meters per second of a surface current. The particles descending from above enter this near still layer and settle nearly vertically for the final stretch of their descent. Their horizontal drift, accumulated over weeks of passage through the water column above, is small relative to the distance they have traveled vertically. They arrive on the seafloor not far in horizontal terms from the point on the surface directly above, a fidelity to origin that the tracking instruments needed to measure it would find almost impressive, except that it is simply physics. Dense particles fall. The water through which they fall is nearly still at the bottom. They arrive below where they formed. The brittle stars intercept some of these arriving particles. The sea cucumbers process others from the sediment where they have settled. The polycades draw some down into the sediment column and distribute them into the deeper layers. The bacteria work on all of it, on the particles still suspended in the water, on the particles that have settled on the sediment surface, on the particles that have been buried by biological activity or by the slow accumulation of new material from above. The carbon in those particles is conserved by this system, passed from organism to organism, transformed from one chemical form to another, but not lost. What is not permanently buried is eventually remineralized into dissolved inorganic carbon in the deep water, where it joins the vast dissolved pool that circulates through the deep ocean on timescales of centuries to millennia before being returned, eventually, to the surface. Nothing in this system is wasted. Everything is processed. The deep sea floor is not a graveyard in the usual sense. It is not a place where things come to rest and cease to matter. It is a place where slow work continues, where the products of surface productivity are received, transformed, and either returned to circulation or placed into the long-term geological record. The organisms that live here do not experience this as significant. They are simply living, eating, moving, reproducing, dying. The significance is a quality the system has at a scale none of its inhabitants perceive. A brittle star on the edge of the bed extends one arm slightly further into the current. A particle approaches. The tube feet along that arm extend imperceptibly toward it. The particle drifts. The arm waits. The cold water between them carries no sound, no light, no warmth. And then the contact happens. One particle, one tube foot, one transaction in the continuous accounting that has kept this community alive in absolute darkness for longer than there have been cities, longer than there have been farmers, longer than there have been hands to hold a tool. The particle is consumed. The arm draws back slightly, returning to its waiting position. Above, the descent continues, patient and unbroken. More particles form at the surface. They aggregate and begin to fall. They pass through the long middle of the water column, processed from all sides, diminished but not entirely consumed. And the ones that survive, the dense pellets, the mineral-bound fragments, the most resistant organic compounds wrapped in mineral coatings that have protected them through every layer of biological activity above. The ones that survive arrive here, in this cold and absolute dark, where the slow organisms that have always been waiting receive them. A polycate with their bradus draws a thread of pale phytodetritus down through the sediment surface. The material disappears into the sediment beneath. The hole it left behind is small, already filling with the particles continuously settling from the water just above the sediment. In a few hours the hole will be gone, covered over, the surface smooth again, as if nothing had entered it. The worm below will continue its work in the dark below, that smooth surface, and the sediment around it will hold, in its layered chemistry, the story of everything that has ever arrived from above. All the blooms, all the seasons, all the years. The sea cucumber moves on across the floor. The brittle stars wait. The bacteria work through the night, which is to say, through every hour, without distinction, because in this place there is no night and there is no day, and the work continues regardless, patient and complete and wholly sufficient to itself. The water down here is cold enough to feel like the edge of something, not the edge of the world, the edge of warmth, the edge of light, the edge of all the ordinary measures a body uses to know where it is. The temperature hovers near freezing without reaching it, held just above by the slow chemistry of salt. The pressure at 4,000 meters below the surface presses in from every direction at once, 350 times greater than the air resting on skin at the surface, a compression so thorough that the water itself becomes slightly denser, slightly darker, slightly more reluctant to move. No light reaches here. None. The darkness is not the darkness of a room with curtains drawn, or a cave at night, or a street without lamps. It is an absolute geological darkness that has persisted, unbroken, since the seafloor was laid down by cooling rock and the slow accumulation of what fell from above. The abyssal plain extends outward from any given point, without horizon or feature, a landscape larger than all of Earth's continents, pressed flat and joined together, covered in soft sediment that gives slightly underfoot the way wet clay does. The sediment is cold, it is almost perfectly still, and from above, with patient and absolute constancy, the rain comes down. It comes slowly. It has always come slowly. A single particle falling through water, this cold and this pressurized, descends at a pace that makes the movement of a sleeping body seem swift by comparison. A gentle drift, a settling, a long descent through layers of water that are themselves barely moving. The particle arrives at the sediment surface, carrying something it gathered at the top of the ocean. Perhaps weeks ago, perhaps longer, it arrives carrying carbon. The sediment beneath a point of abyssal seafloor is not simply mud. It is time, compressed and accumulated, one thin film at a time. Over the course of 1,000 years, across most of the open ocean floor, somewhere between a fraction of a millimeter and a few millimeters of new sediment accumulates. That is the pace of the record, so slow that a layer the thickness of a thumbnail represents centuries, so slow that a core pulled from the seafloor, perhaps 10 meters long, might contain the compressed record of the last several million years of surface productivity, climate, and change, stacked in a sequence so orderly that a trained eye can read it the way a careful reader moves through a handwritten page. The uppermost layer of that sediment is different from the rest. It is softer, paler, less consolidated, a thin blanket of material so recently arrived that it has not yet been compressed or mixed thoroughly into the sediment below. This layer is sometimes called the fluff, or the phytodetritus layer, and it holds within it the most recent delivery from above. Fragments of diatom shells, still recognizable in their geometry, compressed fecal pellets from copepods that lived and died within the past season, organic films and mucus threads that have somehow survived the long descent without being entirely consumed. It smells, in whatever way chemistry smells in cold, dark water, of the surface. It is the surface's most recent letter, arriving after a journey of weeks or months through the water column, written in the particulate language of bloom and death and descent. Below the fluff, the layers darken and compact. The pale bands where they appear are records of diatom blooms, years when the surface was productive, when the spring bloom was thick and fast, and the marine snow fell heavy and white through the water column, delivering more carbon, more silica, more organic matter than the bacteria of the midwater could fully consume on the way down. The darker bands between them correspond to years or decades of lower productivity, when the surface was warm and still, and the marine snow was thin, and what arrived at the seafloor was sparse and already heavily degraded. Between these bands, in the thin alternating layers, are the signatures of climate. A researcher working with a sediment core holds 10 million years of ocean history in a tube that fits in both hands. The oldest material at the bottom of the core was surface water, when the continents were in slightly different positions, and the ocean currents ran on slightly different tracks, and the climate operated under conditions no human ever experienced directly. The carbon in those deep layers was fixed by phytoplankton that lived and died millions of years before the first human ancestors stood upright on dry land, organisms that have left nothing behind but their chemistry and the faint geometry of their shells, compressed into a record that reads, to someone who knows how to interpret it, as clearly as weather data. That is the gift of the seafloor's patience. Everything that falls eventually arrives. Not all of it, most is consumed long before the bottom, but enough, and persistently enough, and in sufficient variety that the sediment accumulates not just as mud, but as information. The carbon that arrives and is buried there does not return to the atmosphere quickly. It does not cycle back into the surface ocean within months or years, depending on where it is buried and how deeply it is incorporated into the sediment. It may remain sequestered for thousands of years, or for millions, pressed gradually deeper by the weight of new accumulation, eventually incorporated into the geological record of the planet itself. Carbon atoms that were part of a living phytoplankton cell at the surface of the medieval North Pacific now rest in sediment on the floor of that same ocean, dissolved into the cold interstitial water between grains, incorporated into the tests of foraminifera, whose shells settled around them centuries after the phytoplankton died. The water surrounding those atoms has not touched the atmosphere since before the first European ship crossed that ocean. It has been circulating in the deep, cut off from the surface, carried slowly by the great sluggish conveyor of ocean circulation that moves bottom water from the poles toward the tropics and back again over the course of 1,000 years or more. Those carbon atoms are not gone. They are waiting. On a time scale longer than all of recorded human civilization, they will eventually be returned to the surface, upwelled and outgassed, and made available again to phytoplankton that will fix them once more and begin the cycle again. But for now, they rest, cold, dark, and still. And then, into this stillness, something large falls. The abyssal plain is not accustomed to large arrivals. The marine snow comes in a continuous thin drizzle, individual particles, individual pellets, occasional fragments of shell or mineral grain sifted down through hundreds of meters of water. The arrival of a whale changes everything within the radius of its chemical signal. A blue whale, the largest animal that has ever lived on this planet, can weigh nearly 200 tons. When one dies at the surface, old or sick or simply worn out, and begins its slow descent, Guinea carries with it a cargo of organic carbon so concentrated in a single point that it represents, for the area of seafloor it eventually settles on, something like several centuries of background marine snow delivery compressed into a single event. The descent of a whale takes days, sometimes weeks depending on the depth of the water and the gases produced by initial decomposition. It arrives at the seafloor already partially transformed, bloated in places where gases accumulated, deflated in others, the flesh around the edges beginning to soften and release the first chemical plumes that will travel outward through the cold bottom water like slow, invisible announcements. The seafloor beneath the arriving carcass has never, in its recent history, experienced anything like this. The thin rain of particles that has been falling for centuries onto that particular patch of sediment has produced a community calibrated to scarcity, organisms built for patience, for finding and extracting the last traces of nutrition from sediment that has already been processed and reprocessed by every previous inhabitant. The carcass changes that completely. Within hours of settling, the chemical signal has traveled far enough to reach the sensory apparatus of animals that have spent their lives moving slowly across a featureless plain, looking for exactly this kind of concentration. The first to arrive are mobile scavengers, sleeper sharks and hagfish, and the amphipods that can detect dissolved organic signals over distances measured in hundreds of meters, their antennae reading the water chemistry as they come. The carcass in these first weeks belongs to movement, to the enormous activity of thousands of individuals feeding simultaneously, stripping the soft tissue from bone with a speed that is utterly unlike the patient, measured pace of everything else on the abyssal plain. In the darkness, with no light to mark the scene, the carcass is surrounded by a dense, churning community that has gathered from the surrounding kilometers of empty seafloor, pulled by the same chemical announcement from every direction at once. After the mobile scavengers have taken what they can, the scene changes. The bones remain. The enrichment phase begins, when organisms adapted to the slower rewards of decomposing tissue, accumulated fat, and the concentrated organic material that has worked its way into the sediment immediately surrounding the bones, establish themselves and persist for months. Polycate worms arrive and proliferate, crustaceans settle. The sediment immediately around the skeleton becomes richer than anything in the surrounding seafloor, and a community of enrichment opportunists, organisms willing to live in dense colonies around a concentrated food source, takes hold with a patience that matches the sediment's own. But the most extraordinary stage comes later. The bones themselves contain lipids, fats stored deep in the marrow and in the structure of the vertebrae, that anaerobic bacteria begin to access after the exterior has been thoroughly processed. In the absence of oxygen, in the cold and pressure of the deep seafloor, these bacteria work slowly and thoroughly, breaking down the lipid reserves and producing as a byproduct hydrogen sulfide. The hydrogen sulfide that seeps from the bones of a decomposing whale carcass is a chemical energy source, and the organisms that evolve to use it, the chemosynthetic bacteria that build their energy budget not on sunlight and not on the rain of organic matter from above, but on the chemical difference between reduced sulfur compounds and the oxygen dissolved in the surrounding water, arrive, colonize the bone surfaces, and establish the foundation of a community unlike almost anything else in the deep ocean. The mussels come, small at first, then in greater numbers, their shells dark against the pale bone surface, filtering the water and drawing nutrition from the chemosynthetic bacteria living in specialized tissues within their own bodies. The tube worms follow, their bright red plumes extended into the water above the bone, the red color carrying hemoglobin built not to deliver oxygen to muscles, but to carry the hydrogen sulfide to the bacteria within them. In the total darkness of the abyssal plain, on what was once the skeleton of an animal that breathed air at the surface and sang in frequencies that traveled hundreds of miles through open water, a community of organisms that has never required sunlight or the ocean's surface productivity is living, feeding, reproducing, and dying. This community persists for decades. A single whale carcass in deep enough water and cold enough conditions can sustain chemosynthetic life for 50 or 60 or even 80 years, the bone structure slowly collapsing inward as the lipid reserves are exhausted and the bacterial communities thin and the muscles decline and the sulfide flux slows to nothing. By the time the last trace of the whale's organic contribution has been consumed and the bones have been incorporated into the sediment, centuries may have passed on the seafloor, time enough for the surrounding plain to have received, through its thin and patient rain, something approaching the same total carbon the whale delivered in its single spectacular arrival. The seafloor keeps its record of both. The thin laminations of background marine snow, layer on layer, mark the steady passage of productive seasons at the surface. The occasional deeper disturbances, the sites of whale falls, the patches where organic matter arrived in concentrated pulses, are written differently in the sediment's chemistry and the preserved shells of organisms that only appear where sulfide was once present. Both are part of the same archive. Both record the same truth, that what lives at the surface of the ocean determines in the end what happens in the dark below. The temporal lag between those two worlds is one of the abyssal seafloor's most striking qualities. A bloom of phytoplankton that peaked in March at the surface of the North Atlantic will not be felt as a pulse of arriving phytodetritus on the deep sea floor until May or June or perhaps later, depending on the depth of the water and the speed of sinking particles and the degree to which bacterial consumption has slowed the descent. The seafloor community is always responding to a past it cannot observe directly. It has no access to the surface. It has no way of knowing whether the signal it is receiving corresponds to a small bloom or a large one, to a warm year or a cold one, to conditions that have since changed or to conditions that are still ongoing kilometers above. It can only read what arrives, in the sequence it arrives, and respond accordingly. The organisms of the abyssal plain have calibrated themselves to this lag. Some benthic species show reproductive pulses that correspond not to the current season, but to the season of maximum surface productivity, weeks or months before. Their bodies in the chemical darkness, reading the arriving organic signal as a seasonal cue and timing their spawning to the abundance it indicates. The phytodetritus that settles on the sediment surface in early summer is to the animals living there the signal that winter has ended. Their spring is not Earth's spring. Their spring is the delayed echo of it, arriving long after the ice has melted and the bloom has peaked and the surface has already begun its slow return to stratification and nutrient depletion. The sediment itself extends this delay further. Material incorporated into the top centimeter of sediment may be reworked by burrowing organisms over the course of years, mixed and redistributed so that a signal from one season is blended with signals from adjacent seasons, smoothed into an average that loses the sharpness of any individual bloom event, but retains the long-term trends. Deeper material, below the reach of active burrowing, is less disturbed. The layers below the bioturbation zone preserve finer resolution, individual bloom signals still distinct in their chemistry, their isotopic ratios, their species composition. Reading the sediment at depth requires careful disentangling of these layers of mixing and preservation, an interpretive process that takes years of laboratory work and returns something extraordinary, a window into the past that no surface observation could provide. Consider a layer of sediment at a depth of 2 meters in a core pulled from the abyssal Pacific. 2 meters of accumulation at the rates typical of the open ocean might represent 100,000 years of deposition. The carbon preserved in that layer was fixed by phytoplankton during a period when the ice sheets of the last great glaciation were still advancing, when sea level was lower by several hundred feet, when the geography of coastlines and the pattern of ocean circulation were measurably different from today. The diatom shells in that layer, still readable under a microscope despite the time elapsed, are species that flourished under those conditions: cooler surface temperatures, different nutrient distributions, different upwelling patterns driven by wind fields that blew across a world no human being remembers or could fully reconstruct from any other source. That layer is quiet now. It holds its record without interpretation, waiting for the tools and the questions that will read it. Somewhere above it, closer to the surface of the core, other layers hold the records of other periods, warmer intervals, cooler intervals, the signature of the Younger Dryas cold reversal, the shift into the current interglacial, the last several centuries of industrial carbon loading, visible in isotopic ratios that change in ways the preceding hundreds of thousands of years never showed. The sediment does not distinguish between these events as significant or trivial. It simply records, patiently, at the pace of millimeters per thousand years. The cold that keeps the abyssal plain at near freezing temperatures is not local. It originates in the polar regions of both hemispheres, where surface water chilled by contact with ice becomes dense enough to sink and spread across the floor of the ocean in slow, thick sheets. This thermohaline circulation, the great overturning of ocean water driven by differences in temperature and salinity, moves bottom water from the poles toward the tropics at a pace so slow that water formed at the surface near Antarctica may not reach the deep equatorial Pacific for more than 1,000 years. While it travels, it carries dissolved oxygen replenished at the surface, oxygen that keeps the deep sediment environments from becoming completely anoxic, that sustains the benthic communities living in the cold, and that allows the bacterial decomposition of arriving organic matter to proceed aerobically in most regions of the abyss. In places where the thermohaline circulation is sluggish, or where the production of organic matter arriving at depth is so high that the oxygen is consumed faster than it can be replenished, the sediment becomes anoxic, devoid of oxygen, and the conditions for preservation improve dramatically. In anoxic sediments, organic matter is not consumed by aerobic bacteria. It persists. The fine details of organisms that fell to the seafloor thousands or millions of years ago are preserved in the sediment with extraordinary fidelity, their cellular structures still visible in their chemistry, their carbon still intact in forms that geologists can eventually read as oil and gas, after millions of years of pressure and heat, have transformed them further still. The deep ocean is, in this sense, both a consumer and a preserver. Most of what arrives is consumed, remineralized, respired, returned to the water as dissolved inorganic carbon that will eventually, over centuries to millennia, make its way back to the surface and out into the atmosphere. But a small fraction, in the right conditions, is kept hidden, incorporated into sediment and then into rock and then into geological formations that persist on timescales that dwarf even the deepest resolution of any sediment core. The carbon in an oil deposit was surface carbon, once fixed by photosynthetic organisms in ancient oceans, sunk through the water column, buried in anoxic sediment, and transformed by time and pressure into something that can be burned millions of years later to release the energy of that ancient sunlight back into the atmosphere in a matter of moments. This is the full arc of carbon through the deep ocean, not a loop, but a cycle with branches, most of the carbon cycling back to the surface within years or decades, a smaller fraction sequestered for thousands of years in the deep water and sediment, a smaller fraction still preserved in geological time, waiting in rock and sediment for conditions that will never come, or for a process that will take longer than the history of life on this planet has yet lasted. The abyssal plane holds all of these timescales simultaneously in a single core pulled on a gray afternoon from cold polar water. The top centimeter holds last season's phytodetritus, still soft, still pale, still carrying the chemical signatures of the specific bloom that produced it. A few centimeters below, the sediment from a century ago has been gently mixed and compressed, but still holds the species composition of a different ocean, slightly cooler, slightly more productive in certain regions before certain changes in the surface had propagated to this depth. And at the bottom of the core, in layers that took the pull of a winch several hours to retrieve from the seafloor, carbon atoms that were last in the atmosphere before the Great Glaciation began lie quietly in the compressed remains of organisms that lived their entire lives in a world that no longer exists. The seafloor receives all of it. It does not hurry any of it along. It keeps the record with a patience that has no equivalent in human experience or in human language, the patience not of waiting for something to happen, but of simply being the place where things arrive and settle and remain. A single carbon atom fixed by a phytoplankton cell during the spring bloom of the year 1200 in the surface water of the North Pacific followed its host cell into the marine snow. When the bloom collapsed, it descended through the twilight water, through the cold and the dark, and arrived at the seafloor after a journey of several months. The bacteria that covered the aggregate it rode had consumed much of its neighbors, but this particular atom was incorporated into a fragment of a diatom frustule, the silica shell that encased the phytoplankton, and because it was bound within the shell's organic lining, rather than freely available on the surface, it was partially protected from immediate consumption. It settled into the sediment. It was covered, over the following centuries, by the accumulating layers of later arrivals. It is there now, approximately 8 centuries later, held in cold sediment under several hundred kilograms of accumulated material, surrounded by water that has not seen the surface since before the first European ships reached that ocean. It will remain there, in all likelihood, for a very long time, not forever. Nothing is forever in the ocean's accounting. The deep water will eventually upwell. The sediment will eventually be subducted at a tectonic boundary, or scraped into an accretionary wedge and folded into rock, and eventually, on timescales of tens of millions of years, exposed again at the surface by erosion and weathering and the slow rearrangement of continents. The carbon will be returned. It always is in the end. The ocean has been running this cycle for hundreds of millions of years, storing carbon and releasing it, burying organic matter and exposing it again, the rate and balance shifting with climate and biology and tectonics, but the fundamental process, persisting, patient, and recursive, since long before the first multicellular organism appeared in the warm, shallow seas of an ancient world. The fluff layer on the sediment surface right now, in this year, in this season, is the newest entry in a record that has been accumulating without interruption for longer than there have been fish in the ocean. It is soft, it is pale, it is full of the chemical signatures of a bloom that peaked weeks ago at the surface, in water lit by a sun that the sediment below it has not felt in millions of years. The organisms living in and just above it are consuming some of it, drawing nutrition from the most recently arrived material, the way a reader might open the latest letter in a long correspondence. The rest will be incorporated, compressed, and added to the record, and the rain continues. It does not stop for the winter, though it thins. It does not stop for the night, though it slows when the surface bloom fades. It does not stop when the water is cold, or when the currents shift, or when the surface world that produces it is going through a period of scarcity. Slower, sometimes. Thinner, sometimes, but continuous, patient, falling through kilometers of dark water at the pace of a thought, fading into sleep, and arriving, grain by grain and flake by flake, at a seafloor that has been receiving it for longer than the mountains have stood, adding each new layer to a record, so deep and so quiet and so complete that it holds in its compressed cold darkness the entire history of the ocean's long, patient breath. The carbon rests, the sediment holds it. The cold water above it moves in slow circuits that will not complete for another 1,000 years. And somewhere far above, in water lit by a sun that has just now risen over a calm and ordinary sea, the first cells of a new bloom are beginning to divide. In the North Atlantic, in early March, the water is still cold from winter. It is gray at the surface and darker below. The light that reaches down through the first few feet is thin and diffuse, the kind of light that illuminates without warming, that reveals shapes without colors. Beneath it, the water column stretches downward for more than 2 miles, and all through that depth, the ocean is quiet and slow and still waiting. Then the days lengthen by a few minutes, then by a few minutes more. The angle of the sun shifts, almost imperceptibly at first, and the light that falls on the surface is a little stronger in the afternoon than it was the week before. Something stirs. In the uppermost layer of the water, where the sun can still reach, single-celled organisms that have been drifting through the dim winter months in low numbers begin to divide. 1 becomes 2, 2 become 4. Within days, in patches that slowly spread and merge and thicken, the surface layer of the North Atlantic fills with green. The bloom does not announce itself. It simply accumulates. The water that was gray last week is now gray-green, and the water that was gray-green this morning is a deeper, richer color by afternoon, and by the following week, the surface has turned the color of shallow coastal bays in summer, dense, alive, almost opaque with life. Beneath it, looking upward through a few meters of water, the light that filters down has the quality of sunlight through frosted glass, broken into shafts and columns that shift with the surface movement, abundant and then spare, present and then briefly withdrawn. Through this shifting light, the first aggregates have already begun to form. They are barely visible. Each one is a loose cluster of dead cells, shed fragments, the sticky invisible threads of exopolymer gel, binding pieces together that would otherwise drift apart. A particle forms in the morning and begins to sink. Another forms beside it and follows. By the time the day has ended and the surface has grown darker and the bloom has pushed upward toward its peak, there are thousands of these particles beginning their descent through the green-lit upper water, each one moving at the pace of settling dust in a room where no one has opened a window. This is how the ocean breathes in spring. Not all at once, not uniformly, not on any human schedule, but at a particular latitude, in a particular year, in response to a particular arrangement of light and cold and the slow shoaling of the winter mixed layer. The ocean exhales, and what it exhales is carbon. The same surface, 3 months later, looks entirely different. The bloom has peaked and faded. The nutrients that fed it, nitrogen, phosphorus, silic acid, have been drawn down by weeks of cell division, and the water, though still warm and brightly lit, has grown clear again. The deep blue of midsummer ocean is a beautiful color and a quiet one. It holds less life near the surface than it did in April. The light passes all the way through it without encountering much to scatter or absorb it. The marine snow that fell so heavily in spring has thinned to a sparse, occasional drift, and the sea floor below is already beginning to process the last of what arrived. The same water, the same depth, a different world. The North Atlantic is only one ocean, and spring is only one season. The ocean's breath is not a single event in a single place. It is a continuously shifting arrangement of pulses and quiet intervals, of regions that never stop producing and regions that barely begin, of surfaces that look one way in winter and another way entirely in summer. Understanding how the deep ocean feeds requires understanding this variation, not as an exception to a rule, but as the rule itself, played out differently at every latitude and in every month across all the ocean basins at once. At the highest latitudes, where the Arctic and Antarctic ice extends across the surface in winter and retreats each summer toward the poles, the ocean's most productive blooms occur not in the open water, but at the edge of the receding ice. As the ice pulls back across the water in spring, it leaves behind a very particular condition. The meltwater is cold and fresh. It sits lightly on the surface, separated from the denser salt water below by a sharp density gradient that resists mixing. Above this stable, shallow, well-lit layer, the sun has risen high enough to deliver real energy. Below the ice edge, nutrients from winter accumulation are present in abundance. And the cold temperature itself, which might seem like an obstacle to growth, turns out to be an advantage for carbon export. In cold water, bacteria work slowly. The organic material of dead cells and shed fragments does not remineralize as quickly as it does in warmer seas. The sticky exopolymer particles aggregate more readily. The fecal pellets of krill and copepods, dense and compact, sink through the cold water in near straight lines, carrying their cargo of packaged carbon downward with unusual efficiency. Where a marine snow particle might be half consumed by bacteria before it reaches a few hundred meters depth in a tropical ocean, the same particle sinking through polar water may survive much further into the water column before the bacteria have worked through it. At the edge of the Antarctic ice, in the brief and brilliant Southern Ocean summer, this efficiency reaches its peak. The water is a green, so saturated it approaches black at depth. Krill aggregate in swarms that can stretch for miles, grazing on the dense surface phytoplankton and producing pellets that descend quickly through the cold, clear column below. The ice edge itself is a hard white boundary, on one side flat and featureless and barren of surface life. On the other, within meters, a surface so dense with cells that the water has changed color. The transition is abrupt enough to be visible from the air. Below that green surface, the particles fall. They fall through water that is just above freezing. They fall through the last faint traces of sunlight at 200 feet, then into complete darkness below, and they continue falling through a cold and pressured dark that does not vary much with depth, that does not brighten or warm at any point in its long descent, for the better part of a mile before reaching the seafloor. And because the water is cold, and because the bacteria work slowly, a greater fraction of what left the surface arrives. The polar ocean sends more of its carbon to the deep than most other regions of the same size, and the deep seafloor below it is accordingly richer. This variation between the productive and impoverished, between the cold and efficient, and the warm and active, is not a flaw in the ocean system. It is the system. The ocean does not produce uniformly, because it cannot. The conditions for marine snow formation are local, seasonal, and responsive to forces that operate at scales from the daily turning of the Earth to the multi-year oscillations of global climate. Every surface is different from every other surface, and the seafloor below it carries the record of that difference, layer by layer, season by season, for as long as the sediment has been accumulating. The most extreme version of this variation sits at the other end of the productivity spectrum in the subtropical gyres. These are the great circular current systems that occupy the broad middle of each ocean basin, the North and South Atlantic gyres, the North and South Pacific gyres, and the Indian Ocean gyre. Together, they cover roughly 4 out of every 10 square miles of the ocean's surface. They are by area the largest biomes on Earth. They are also very nearly deserts. In the center of a subtropical gyre, the water is the color of deep sky. On a clear day, sunlight penetrates 50 or 60 feet before the water dims to a rich blue-black. There is almost nothing to scatter the light, because there is almost nothing there. The surface layer has been warm and still for so long that the nutrients that once existed in it have been consumed and settled, and the density difference between the warm surface and the cold water below is strong enough to resist the mixing that might bring fresh nutrients up from depth. The phytoplankton that live here are extremely small, specialists in survival under scarcity, not in growth under abundance. The marine snow in a subtropical gyre is sparse enough that sediment accumulates on the seafloor below at rates that can be less than a millimeter over 1,000 years. A layer of sediment, a centimeter thick, may represent 10,000 years of slow, patient deposition. The organisms on the seafloor below a gyre center live in a kind of permanent lean season. They eat little, grow slowly, and wait with the patience that deep time alone can impose. And the same ocean surface, a few thousand miles to the east, in the waters off Peru and Chile, and the coast of California, presents an entirely different face. Here, the wind drives the surface water offshore, and cold, nutrient-rich water rises from depth to replace it. This process, called upwelling, delivers to the surface exactly what the subtropical gyres lack: deep, cold water loaded with the nitrogen and phosphorus that phytoplankton need to grow. In an upwelling zone in a good year, the water near the coast runs green and thick with life. Anchovies gather in vast schools. Seabirds work the surface in numbers that make it look crowded. The marine snow forming below falls fast and thick, and the carbon export to the seafloor is among the highest measured anywhere in the ocean. This richness, though, is not stable from year to year. It depends on the wind, and the wind in the tropical Pacific is controlled in part by a pattern of ocean temperature and atmospheric pressure that shifts on a cycle of several years, irregular in timing and variable in strength, with consequences that reach every ocean on Earth. In years when the eastern tropical Pacific warms anomalously, the ocean temperature rising several degrees above normal, along a broad band of water stretching from the coast of South America westward toward the central Pacific, the winds that normally push surface water offshore weaken, and the upwelling along the coasts of Peru and Ecuador slows or stops. The deep, nutrient-rich water stays down. The surface warms, the phytoplankton thin, the anchovies disappear into deeper, cooler water, or simply decline in number. The seabirds fail to breed, and the marine snow, which had been falling thick and fast toward a seafloor already accustomed to abundance, thins to a quiet trickle. This is the condition known as El Niño. Its effects are felt not only in the waters where it originates, but across the global ocean, through shifts in rainfall, wind, and ocean temperature that propagate outward from the tropical Pacific, and alter conditions in the Indian Ocean, the North Atlantic, the monsoon systems of Asia and Africa. A strong El Nino year leaves a signature not only in weather records and crop yields, but in the sediment traps deployed in the deep ocean and in the living communities of organisms on the seafloor that must process less carbon than they did the year before. The sediment record below a region affected by El Nino cycles carries those cycles in its layers. A chemist extracting a core from the seafloor of the equatorial Pacific and measuring the abundance of organic carbon from layer to layer can read upwelling strength backward in time through thousands of years. The cycles of El Nino and La Nina are written in the sediment as surely as they are written in the growth rings of trees or the ice cores drawn from glaciers. The ocean does not only respond to climate, it records it. La Nina brings the opposite condition. The waters of the eastern tropical Pacific cool below normal. The trade winds strengthen. The upwelling accelerates along the coast of South America and through the central equatorial Pacific. The phytoplankton return in abundance. The marine snow thickens. The seafloor communities that had been living lean receive their replenishment, and the particle flux records a pulse that corresponds precisely to the anomalous productivity at the surface, delayed by the weeks it takes for those particles to sink through the water column. This delay, the lag between surface event and seafloor arrival, is one of the most quietly disorienting aspects of the deep ocean's relationship with the surface above it. The seafloor is always receiving the past. In shallow deep-sea settings, a few hundred meters of water, that past might be days or a few weeks distant. In the abyssal ocean, below 4 or 5 kilometers of water, the particles that arrive in a given week left the surface months before, sometimes more than a year before. The surface spring bloom that drives a pulse of fresh phytodatus to the deep-sea floor may have already given way to the quiet of midsummer before the first particles from that bloom reach the bottom. The organisms on the seafloor respond to a season that from any surface perspective is already over. When fresh phytodatus arrives after the long winter to spring transition, the sea cucumbers begin moving more actively. The brittle stars extend their arms more fully into the current. The polychaetes come to the surface of their burrows. The physiological trigger for these responses is not light, which they cannot sense. It is chemistry, the particular combination of compounds that arrive with freshly settled phytoplankton, the sharp ratio of organic carbon to nitrogen that signals material not yet fully degraded. The deep ocean community knows spring by the smell of it, arriving weeks late from a world they have never seen. The Arabian Sea experiences this seasonal signal in a particularly clear and dramatic form. Twice each year, the reversal of the monsoon winds drives intense upwelling events along the coasts of Oman and Somalia. The southwest monsoon, arriving in summer, pushes the warm surface water offshore and draws cold, nutrient-laden water up from depth. The phytoplankton respond within days. The bloom can be seen from satellites as a spreading green stain across the surface of the northwestern Indian Ocean. Below it, the particle flux to the deep increases sharply, and in sediment cores extracted from the seafloor of the Arabian Sea, these monsoon-driven pulses are preserved as distinct layers, slightly darker bands of organic-enriched sediment alternating with lighter, leaner intervals, stretching back through thousands and then hundreds of thousands of years. Oceanographers studying these cores are not simply reading the recent past. They are reading the history of the monsoon itself, preserved by the ocean with the same patience and regularity that the ocean brings to everything else it does. The intensity of the summer monsoon over the Indian subcontinent 10,000 years ago or 50,000 years ago, or 200,000 years ago, is encoded in the sediment of the Arabian Sea, not as a statement, but as a physical fact, a slightly thicker layer, a slightly higher concentration of the silica shells left by diatoms that bloomed in the upwelled water. The ocean does not interpret its own history, it simply stores it. This capacity for storage, for the accumulation of physical record across timescales that dwarf any human system of measurement, is inseparable from the process of marine snow itself. Each particle that leaves the surface and survives its long fall through the water column carries a piece of the surface world with it. Not a memory, it has no memory, but a chemistry, a structure, a ratio of elements that reflects the conditions of its birth. A diatom frustule, the intricate silica shell of a single-celled algae, arrives on the seafloor with its geometry intact, its species determinable, its age measurable. It was assembled at the surface by a cell that lived and died in a week or two, in a particular ocean, in a particular season, under particular conditions of temperature and nutrient availability. And then it sank, and the cell that built it decayed away, and the shell alone arrived on the seafloor and was buried beneath the next thin layer of settling material. Billions of these shells, falling continuously over millions of years, have built up the deep-sea sediment layers that now hold the most detailed record of past ocean conditions available to science. The frustule that rests in a sediment core extracted today arrived from a surface that existed in a climate the Earth has not experienced since. The diatom that built it has no descendants alive today because the species evolved and changed, but the shell it left is still there, a few centimeters below the surface of the sediment, intact and datable, waiting with the patience that the deep ocean specializes in. The deep ocean specializes in patience because it has no choice. Below the first few hundred meters of water, the forces that create urgency at the surface, the cycle of day and night, the swing of seasons, the surface turbulence of weather, do not reach. The water is cold and dark and still. It moves, but it moves slowly, on time scales of decades to centuries to millennia, driven by the density differences between polar and tropical water, between warm and cold, between fresh and salt. A water mass that sinks in the North Atlantic near Greenland, cooling and freshening and growing heavy enough to slide down and move southward along the deep ocean floor, will travel through the Atlantic, around the tip of Africa, and into the Pacific and Indian Oceans over the course of roughly 1,000 years before it finds its way back to the surface. In that 1,000 years, the carbon dissolved in it will have been out of contact with the atmosphere, isolated from the carbon cycle of the surface world, settling slowly through a dark interior that has not changed much in all the centuries of that transit. Carbon that sinks into the deep ocean and stays there is carbon that is not in the atmosphere. The biological pump, the process of photosynthesis at the surface, followed by aggregation and the long fall of marine snow, followed by consumption and burial, moves carbon from the air into the deep sea, where it remains for timescales that range from decades to millions of years, depending on how deep it goes, how efficiently it is buried, and how the ocean's circulation eventually returns it to the surface. The quantity moved each year is small relative to the enormous store of carbon already held in the deep. But across the timescales at which the Earth's climate operates, across thousands and tens of thousands of years, that small annual movement is a primary control on the concentration of carbon dioxide in the atmosphere, and therefore on the temperature of the planet. During ice ages, when the global ocean was colder and more stratified, The biological pump appears to have operated more efficiently, drawing down more carbon dioxide from the atmosphere and helping to keep global temperatures low. During interglacial periods, when the ocean warmed and the pump became less efficient, atmospheric carbon dioxide rose and temperatures climbed. The ice cores drawn from Greenland and Antarctica preserve this connection with extraordinary clarity. Layers of ancient ice that hold trapped bubbles of past atmosphere, their carbon dioxide concentrations rising and falling in near perfect synchrony with the temperature records preserved in the same ice. The ocean's slow breathing, the marine snow falling year after year into the dark, is part of what shaped those swings. The surface of the ocean in any given week reflects all of this. It reflects the season because the season controls the light and the mixing depth. It reflects the latitude because latitude controls the nutrient supply and the temperature and the character of the plankton communities. It reflects the last few years of interannual variability because the anomalies of temperature and upwelling that come with changing climate patterns are written in the populations of organisms still present or absent from the bloom, and it reflects in a diffuse and subtle way the long history of the ocean's own evolution, the slow accumulation of species and ecological relationships that have shaped how efficiently the surface transforms sunlight into carbon and how reliably that carbon falls. A ship crossing the subtropical Pacific in summer, far from any coast, is crossing a surface that seems empty. The water is warm, the blue is deep and unclouded. No seabirds follow the wake for long because there is little for them to eat. Below the hull, the marine snow is sparse, a particle every few feet in the upper water, growing sparser with depth, the occasional dense pellet from a copod falling faster than the rest. The seafloor below, more than 3 miles down, receives this trickle with the same patience it has brought to the task for millions of years. A ship crossing the subpolar North Atlantic in April is crossing a different world. The water is cold enough to be uncomfortable and green enough to look shallow, even where it is hundreds of feet deep. The hull passes through patches of foam where the surface concentration of organic material is high enough to make bubbles that persist. Below, the marine snow is falling in something closer to curtains than flurries, dense aggregates, fast-sinking pellets, the first phytodetritus of the spring bloom, still fresh and pale as it passes through the first few hundred feet. The seafloor below, where brittle stars have been raising their arms into the current for weeks in anticipation of exactly this, will receive its annual pulse within days. Both of these are the same ocean system, viewed from different places at different times. The same physics governs both. The same organisms, phytoplankton, zooplankton, bacteria, are present in both, in wildly different proportions. The same carbon cycle runs through both. But what the deep ocean receives from each of them is so different that the communities below them have evolved to live in different conditions of abundance and scarcity, of episodic pulse and continuous trickle, and have built different strategies for surviving the intervals between deliveries. The differences between ocean regions are not fixed. They shift with climate on every timescale, from seasonal to glacial, and the marine snow system shifts with them. As the ocean warms, the stability of the surface layer increases in many regions, suppressing the mixing that brings nutrients to the surface and reducing the intensity of blooms. In polar regions where the sea ice is retreating earlier and forming later, the timing and character of the ice edge blooms are changing. The species composition of the phytoplankton communities is shifting, with smaller cells increasingly dominant in warming waters, and smaller cells produce smaller particles that sink more slowly and are more likely to be consumed before they reach depth. These changes matter because the biological pump is not a given. It operates at the scale it does because of a specific arrangement of ocean temperature, circulation, nutrient supply, and species composition that has existed in broadly similar form for millions of years. Disruptions to any of these conditions ripple through the system, and their effects take decades or centuries to work through the ocean's vast interior. The deep sea floor is not yet receiving the full signal of the surface changes that are currently underway. What arrives in the coming centuries will carry the chemistry and composition of a warming ocean, smaller particles, altered nutrient ratios, different species. The sediment layers that form from those particles will be readable, eventually, by researchers who will identify them as the marker of a particular period in the ocean's history, the same way that the Ice Age layers are readable now. None of this changes what is happening tonight, in every ocean basin, at every latitude, in the dark below the surface. Marine snow is falling. It is falling slowly, as it always has. In the high latitude seas, where the spring bloom has come and gone, and the summer phytoplankton are still dividing in the long northern light, dense pellets from krill and copepods are descending at 100 or 200 feet per day through cold and pressured water. In the equatorial Pacific, sparse and occasional aggregates drift downward through water that has been warm and nutrient poor for months, their progress so slow that they may spend weeks crossing a few hundred feet. In the Arabian Sea, the particles from the retreating southwest monsoon bloom are still arriving at depth. The seafloor communities there already reading the chemical signal of a surface event that ended weeks ago. All of this is simultaneous. All of it is continuous. In the deep Atlantic, a water mass that sank from the surface near Greenland 1,000 years ago is still moving slowly southward along the seafloor, carrying carbon that was dissolved at the surface during the medieval period, the composition of that water reflecting the ocean conditions of a world no longer reachable except through records like this one. In the deep Pacific, sediment layers that formed during the last glacial maximum are being slowly buried beneath the sediment of warmer interglacials, and the chemistry of those layers holds a record of upwelling strength and productivity and carbon export that will be readable for as long as the sediment remains undisturbed. In the abyssal plains, in total darkness, under pressure that would close a hand around any surface world object like a fist, the slow rain continues. It arrives without drama. A single particle, pale and loosely structured, smaller than a grain of table salt, settles toward the sediment surface and comes to rest on top of the layer that arrived the day before. No organism witnesses this moment. No one is counting. The particle simply arrives, as particles have been arriving here for tens of millions of years, and adds its small weight to the accumulation of everything that has fallen before it. A sea cucumber moves across the sediment nearby at a pace that cannot be measured over the course of an hour, but that is, nonetheless, movement, a slow continuous advance through sediment that has already been processed by generations of its kind. It draws the sediment through its body. It extracts what little nutrition remains. Behind it, the sediment is slightly rearranged, the layers slightly blurred, the carbon that arrived from the surface months ago converted into the tissue of a living animal in the dark. The particle that arrived a moment ago will be buried within days. The sediment grain that arrives beneath it tomorrow will press it down a fraction of a millimeter